Hello and welcome to Epiphytic cacti. Today we're going to take a closer look at the surface of Rhipsalis. I read this paper recently, this stem anatomy of Rhipsalis cactaceae and its relevance for taxonomy. This is by Alice Calvente, Regina H.P. Andreatta, and Ricardo C. Vieira. What they did in this paper was they actually looked at the epidermis of Rhipsalis serioides, elliptica, grandiflora, paradoxa, pentaptera, and teres. Apparently, this is something that could potentially work very well for the identification of Rhipsalis. And this got me really curious. So I decided to get a microscope, get some materials, and start looking at the epidermal layers of my Rhipsalis. There are a few ways to do this. One way is to take an epidermal peel. It is quite complicated just at home to peel off the layer of the epidermis of a cacti. For Rhipsalis, it's a little bit difficult, particularly when you're thinking about something along the lines of like Rhipsalis clavata. Some of the larger flat-stemmed Rhipsalis, it's easier, but you really can't get any of the plant tissue in the way, or you can't really see what you are looking for when you take an epidermal peel. This is an example of what an epidermal peel would look like from a Rhipsalis. You can see all these kind of funny eyeball looking things everywhere here. See the little eyeball looking things? Those are actually called stomata. And it's really the pore on the surface of the cacti that is allowing it to breathe. So basically their little mouth is all over. And that's a little bit freaky to think about, but that is what is on the surface of plants. You can also see the kind of coloration around the stomata. There's like a little bit of green, and that's what happens. That's chlorophyll. That's what happens when you get a little bit of the plant tissue. And if you get too much of it, you really can't see it. So you can look at these very, very magnified like this, and you'll still sort of be able to see like the differences and stuff. You'll really be able to kind of be like, oh, you know, something about this looks different. But I actually found that it was a little bit easier to see the differences if you looked at it from less magnification. So basically I found it easier at 40x. Here you can see different views. One of the plants is just really fun. This one here, which would have had those betulins, it was a Rhipsalis elliptica that was very, very, very intensely red on the surface. And you know what's really funny? That red coloration is truly only skin deep. When I took the epidermal peel, I kind of laughed because I've never really damaged one of those plants before to even notice it. But I took an epidermal peel with a razor, and when I did it, you could actually see the green tissue underneath just the red surface. So it was kind of funny. It truly is only skin deep. A different method of doing this is to take some kind of clear nail polish and to brush it on the surface of the stem and allow it to completely and totally dry, and then use a piece of clear tape. Box tape is preferable here. And you just lay that down, you pull it up, and then you affix it to a slide and you look at that under a microscope. It is obviously not as detailed, but what you will get is a pretty clear stomatal pattern to be able to look at. You'll be able to see the stomata, you'll be able to see the density, you'll get a general idea. And so that's how we chose to do it, and for good reason. I wanted them all to be exactly the same, and some of my plants are just not big enough to be able to take an epidermal peel without damaging them. So this is also a way that you could do it without causing a whole lot of harm to your plants. I'm sure they don't really like breathing in nail polish, but you know, it's for science. So what we're looking at here is this kind of epidermal impression of, and I'm just going to call them epidermal scans, really. It's at 40x under a microscope. All the photographs are taken in the exact same way, zoomed in to the exact same magnification. So all of these are exactly the same. And what we have here is a single plant, and these are 3 different size stems on the same plant. And I have taken a sample of each one of these. One of them was very small, one of them was kind of a medium size, and one of them was very large. And this is Rhipsalis pachyptera. And you can see that there is some variation in these scans. The stomatal density, so we see their little mouths here, and you can see this one's a little bit less dense and this one's more dense. One thing that can impact that is sun exposure. Air quality is another thing that can affect stomatal density. So a place that has a lot of pollution, you're probably going to have a completely different stomatal density. Rhipsalis don't seem to be super impacted the way that I'm sure that some other species of plants are super impacted by this. Like you can see that there is some clear difference in stomatal density here, but not in a way that I would look at these and be like, they're completely different species. The pattern was always pretty clear, but you could have a little bit of variation here and there. This middle stem here and even the small one are probably stems that didn't even grow in my collection. They probably grew in the collection of wherever this plant came from. Only this one would have grown in my collection. And so it might be that it got a little bit less light. The really large size of that stem might actually cause the stomatal sort of pattern or the surface pattern to sort of stretch out a little bit. It does give you the general idea of how similar they are across the same plant with different size stems. Here we have another example of one. So this one I just call Rhipsalis species Affinis elliptica, meaning that I believe that it has some kind of strong relationship to Rhipsalis elliptica. The scan on these is very different from the scan on the previous plant, indicating that they are in fact different plants. And they look so different that they're probably not even the same species. And once again, you can see here where I took different size stems and looked at them, one very small, one very large, and you see much less variation in this plant than you saw on the last one. And here we go again, we have Rhipsalis elliptica, and this one, again, I took totally different, a little tiny skinny stem, a longer skinny stem, a big fat stem. And again, these are all on the same plant, and you can see that there's not a whole lot of variability there. But between these three plants, again, you can see that Rhipsalis pachyptera definitely stands out as being distinct. These two, they look similar, but not exactly the same. So that's kind of why I think this plant is closely related to Rhipsalis elliptica. Another thing that can be done is that you can actually use a tool like this stomata counter, to try to count the stomata density of the samples that you've taken. So you can see this is at universe.rainbow.com, stomata counter. And then here you can see me passing a sample into this. I did find using the model Stomata Counter 4 yielded the best results here, but you can basically see that it can sort of detect the stomata there and actually count the density. And you might be kind of surprised sometimes, because sometimes it'll look like two of them are really, really similar, but then whenever you pass them through the stomata counter, you can see that like some of them, the stomata is way more dense than you thought. So it's a little bit of an interesting tool. So I wanted to take a second and talk a little bit about Rhipsalis pachyptera. Historically, there has been some confusion within Rhipsalis pachyptera. In the Cactaceae 1923 by Britton and Rose, they depicted two distinct plants as Rhipsalis pachyptera. The two plates shown here is showing the two plants that were depicted. Based on floral characters, fruit, and morphology, these clearly would not have been the same species. I don't believe they are even forms of the same species or subspecies, but actually distinct species. The floral characters of the plant depicted on the right match quite well with the floral characters of the previous specimen, where the epidermal impressions appear to show a stronger correlation with Rhipsalis elliptica than Rhipsalis pachyptera. The floral characters of plants, similar to what is depicted on the left, are quite different. The real issue is what came first under the name Rhipsalis pachyptera. That seems unclear. At the time Rhipsalis pachyptera was described, no illustration was provided with it. As a matter of fact, guys, the description was so sparse, you would have a really hard time identifying any plant based off of the original description. It said very little. One thing that it did say was that the plant had very short flower ovaries, but even that is a little bit confusing because compared to what? It didn't really say. The very first illustration we ever saw of Rhipsalis pachyptera historically, it questioned whether or not what was being illustrated was even the correct plant. So even that we can't rely on. And all following illustrations appear to be confused and depict something similar to the plant on the right. In more recent years, I have seen some depiction of the plant on the left as Rhipsalis robusta, which was reduced under Rhipsalis pachyptera, but can't vouch for the accuracy of that. All the sampled materials I could find of Rhipsalis pachyptera that have been used for molecular phylogenetic studies are similar to the plant on the left. This makes sense considering all these studies have placed Rhipsalis pachyptera as being a close relative of Rhipsalis agadoensis, a species that has a strong morphological, floral, and fruiting similarity to the plant on the left. Some botanists have even synonymized the two species. When a species doesn't appear to conform quite right to the categories provided, we can often see a lot of mislabeled specimen in cultivation. This, of course, makes sense because on some level, we know it doesn't fit quite right. Let's take a look at some of the plants in my collection that look very similar to this plant that is on the left. You can see these floral characters match very, very well, as do some of the characters of the stems. But you can see that large amount of stamen, the long flower ovary. Now let's take a look at this field guide. In this field guide, we can see a depiction of Rhipsalis elliptica out in the wild, and it bears a strong resemblance to the Cactaceae plant that is up in the upper left-hand corner. We can see that the floral characters are very similar. I definitely want to point something out about this plant from this field guide. Take a look at illustration 130 and take a look at the shape of the unripe fruit that was captured of a plant that they are saying is Rhipsalis elliptica. All right, let's look at the same field guide and what they're telling us that Rhipsalis pachyptera is and compare that with the plant that was illustrated with the clusters of the white fruit. We can see in illustration 198 those same clusters of fruit. We can see that there looks like there's some kind of stages of it going from maybe kind of like a pink to a white. So sometimes this might have pink fruit, sometimes it might have white fruit, but note the shape of the fruit. Look at how short and squat it is. Look at the flowers. They don't have that many stamen. They have a lot more petals. If you look at the unripe fruit, you can see, again, very short, very squat looking. If you look at the flower ovary, you can see very short, very squat. The flower bud that is in the upper left and the flower that is in the lower left are actually from a plant that matches very closely the characteristics of the plant that was in the Cactaceae with the red epidermis that was labeled Rhipsalis pachyptera. The plants or the flower bud that is in this flower bud and then this flower right here are flowers and flower buds that match very closely the flowers and flower buds of the plant that was on the left with the white fruit, the one that was probably like Rhipsalis robusta at some point. And then this is a very classic Rhipsalis elliptica. And then this is, of course, Rhipsalis pachyptera. And then this is a flower that very much kind of resembles what was going on in the drawing with the red epidermis. And then we can kind of see the fruit like before it was actually ripe here. And so I'm going to point out a couple of very obvious things about this. The flower tube here, the ovary part of the flower, looks way, way different on this plant than it looks on either of these clones of Rhipsalis pachyptera. Like you can see that, like, yes, this flower ovary is very, very, very short and squat, and this one is a little bit longer. You rarely see Rhipsalis pachyptera with a long flower ovary like that. It's usually quite short like this. But even so, when you compare this flower ovary with this flower ovary, you can see that they are very different. Now, when we compare this one with a completely normal Rhipsalis elliptica, this Rhipsalis elliptica is actually this one. When we compare these, you can see that these two look really, really similar. It's just that one of them has a larger, kind of more robust looking flower, but they are very similar. Based on the flowers, what we see is that there is some kind of relationship here. There's some relation between these plants. And you saw the same thing with the epidermal scans. There appears to be some kind of relation with these plants, a much stronger relationship between them together than there is with them when compared with Rhipsalis pachyptera. So something is very off there. And again, even in this unripe fruit, we can see that these are very different plants. Like I said, there's no part of me that believes that these are the same species. I do want to bring up the fact that this isn't that unusual. So there are other places within Rhipsalis where we see this kind of size thing, where we see a smaller size plant, a smaller size flower, and then a larger plant and a larger flower while still being the same species. One such example would be in Rhipsalis nevis armandii. We have form nevis armandii and form megalantha. They're just forms, so that's not even a subspecies status, meaning that's just some kind of random mutation that's happening within the same population. So it's not that weird to think that Rhipsalis elliptica might have this same kind of odd little random mutation happening where we have smaller plants and larger plants. And so here we see lots of different clones, 4 different clones of Rhipsalis pachyptera in my collection. You can see the floral variations. You can see like sometimes they might have like 5 petals. Depends on how big the plant is sometimes, or just the clone. Like how much energy does it have? What's its care like? So, there's like the little petals that are in the back. So like you can see that little petal right there? Sometimes those petals will develop into full-fledged petals depending on how much the energy the plant has, and sometimes they will stay little tiny sepals and not develop. And so that's why sometimes you can have a Rhipsalis pachyptera that only has 5 petals, but then you can have a Rhipsalis pachyptera that has way more petals. Like I think this one over here on the left, on the far left, has 3, 6, 9. And so typically Rhipsalis pachyptera has more petals, less stamen. They all have these kind of short, squat little ovaries. They all have similar amount of stamen, similar arrangement to the stamen. They kind of bud in a similar way. They form in a similar way. They flower around the same time. Let's take a quick look at what these phylogenetic trees might look like for the species that we're about to look at. We're going to be looking in the subgenus Philarthro-Rhipsalis first. And so you can see that all of these species would have evolved from a shared common ancestor here. And so they would have evolved down in 2 different branches. We have one group, which is the Rosellii triangularis cereoides agadoensis pachyptera grandiflora and pidieri group. And then we have this other larger group. And you can see that within those groups, you can see that they have more evolutionary trees. So Rhipsalis grandiflora and Rhipsalis pidieri would have evolved from the same common ancestor. And there was a split off here where Rhipsalis Rosellii triangularis cereoides agadoensis and pachyptera evolved from the same common ancestor. And then you can see that agadoensis and pachyptera are more closely related than the other ones. maybe there were some question marks that I put on here because I don't technically know exactly where they go. Mostly because there actually isn't a fully and totally complete phylogenetic tree for all the Rhipsalis species. And then we have on the top, we can see that Rhipsalis elliptica and the Macranthas all evolved from the same common ancestor back here. But we can see, of course, that the Macranthas are more closely related. And then we see Rhipsalis gobelliana, rhombia, and ewaldiana form a clade. And we can see that cuneata, subspecies cuneata, subspecies australis, occidentalis would form a clade. Oblonga, Bartholii, and Crispata would form a clade. Then you can see who each other's closest relatives are, Bartholii and Crispata, and, the cuneatas, of course. And you can see that they all kind of have a group here where they evolved from the same common ancestor. So you would expect these guys to, of course, be closer morphologically. And florally, they are quite similar. These are some of the Rhipsalis that I think my personal opinion is that it's actually this group that's really hard to differentiate from each other more than any other Rhipsalis group. And then we have Rhipsalis olivifera, which is kind of out here on its own. So you can see how these would have evolved from the same common ancestor here, but they split off in 3 different ways. And Rhipsalis olivifera is just kind of by itself. Let's kick it off by looking at a very confusing plant. This plant shares a lot of morphological characteristics with Rhipsalis elliptica, but is also very confusing because some of the stems, like these stems here, tend to kind of look similar to some Rhipsalis pachyptera. To make matters worse, the plant produces white fruit under certain conditions, and it produces pink fruit under other conditions. It has rather elongated cylindrical fruit that was very similar to the fruit that we saw in the field guide. And the flowers are those big, fluffy, yellow flowers with the tons and tons of stamen. And the scan of this looks much more like Rhipsalis elliptica than it looks like Rhipsalis pachyptera. So later on, we'll see a ton of slides for Rhipsalis pachyptera, and we'll see just how different these plants actually look. This one was just labeled Rhipsalis species affinis elliptica. I genuinely do think that these are Rhipsalis elliptica. Perhaps there's some kind of thing where maybe there could be forms defined there, or maybe even subspecies. I don't really know. I'm not a botanist. I think that I'm just going off of what these scans look like, and florally and morphologically, like what I know the species to actually be from the experience of growing them. The scan here, when these are compared, you can see that they're very similar. They also have identical looking floral characteristics and fruit, but the fruit on this one tends to ripen to be more pink than it does to white. So these two plants, while they might actually look kind of different just in appearance, they appear to be extremely closely related. This is actually the same plant as this. It looks like I had just picked up cuttings from somewhere else at some point. Then you can see again, we have very similar scans here. And then this is another one that has the same kind of flower. This one apparently came from the Botanical Garden in Berlin. I'm not sure how true that is. That's just what the cutting said when I received it. You can see these scans look a little bit different, but not so different that I would say that they couldn't possibly be the same species. And the floral characters are telling me that they are. You can see the large spray of stamen. And unfortunately, the one thing about this plant is that I did not have any growth from this in my collection to scan. So the only growth that I could scan was the original cutting. And of course, it would have been grown under different conditions, and therefore the scan would probably be different, like the stomatal density might be a little bit different. But you can still kind of make out that they're similar and are probably the same species. I guess florally, I don't really have to guess. I can see that florally, they are the same species. And then we have this guy, which is in cultivation as Rhipsalis rhombia Dave. And again, we can see that these guys, they have similar scans. Now, this one, to me, when I purchased this plant and I looked at it and I examined it up against a Rhipsalis elliptica that is well known that I have, they look the same. They don't really look that different. They are growing in the same way. And this plant actually looks like a lot of pictures I've seen of wild specimen of Rhipsalis elliptica. So this plant is most definitely not Rhipsalis rhombia. And I have seen it in cultivation under tons of different names. So I've seen it as Rhipsalis elliptica, Rhipsalis pachypteriform rubra, Rhipsalis pachyptera, Rhipsalis crispata, Rhipsalis platycarpa. I mean, just a ton of ridiculous names. And I think that the reason why that happens is because it can be kind of weird when the plant is not really conforming to what you think it should be. So when we think of the typical Rhipsalis elliptica, something about this plant is saying it's a little bit different. And actually, if we go to the Tropiflora website and we look at this plant, we can see here they have different pictures. We can see that there are some flower pictures. And here we see these do not look any different from your totally typical Rhipsalis elliptica flowers. There's nothing special or unique about these at all in comparison. They definitely don't look like Rhipsalis rhombia flowers. So kind of confusing as to how this ended up under the name Rhipsalis rhombia here, but morphologically very, very similar to Rhipsalis elliptica that I have seen that have been out in the field, like wild pictured, I guess we would say. And then here we can kind of see this interesting picture, which I think is why this plant fell under so many different names, because I think that what we're seeing right here is a jump in characteristics from a juvenile form of this plant into a more adult form of this plant. And that can be confusing because Rhipsalis elliptica particularly seems to be a plant that will take a really big jump sometimes. Some clones, not all clones do this, but a lot of the clones that I have seen appear to be able to do this, where they have these very small stems when they're young, and then over time, they suddenly start producing stems that are huge, like bigger than your hand huge, and that can be really confusing looking. So if you get cuttings of one of these Rhipsalis ellipticas and they're like huge stems, a lot of people think that it has to be something else, like it can't be Rhipsalis elliptica because it doesn't have huge stems like that. But even the botanical texts and descriptions tell us that yes, they can have humongous stems. So it's kind of interesting. But I wanted to show this picture because it's actually relevant for something that we're going to look at next. But again, here's another one, really not really any different from your typical Rhipsalis elliptica. You can see these elongated stems. You can see how they're kind of like stretched out. In some scenarios, like right here, you can see this big elongated basal stem here. This is a very typical growth habit for Rhipsalis elliptica. So then if we go look here, I actually believe that this is what happens to this plant when it is more in its adult form. And so you can see how it looks different here. And this would be very confusing to people because they would think that this would have to be a different species. But it is still probably just Rhipsalis elliptica. And as a matter of fact, this scan looks very, very similar to just your completely and totally typical Rhipsalis elliptica scans that I took that we'll see in a little bit. And then here we can see another one, and this one is interesting because this is actually just a juvenile form of the same plant. And this came from a totally different source, so it was grown under even more different growing conditions. And you can see that this growth right here, all this growth that we're seeing right here in its sort of juvenile state, we would never really look at that growth and think that that was anything other than Rhipsalis elliptica. Typically, for people who are very into Rhipsalis anyway, you would never look at that and think anything other than that. And it's hard to like reconcile how this plant could be the same as this plant. But you can actually see some of this adult growth coming here. And there you can start to kind of see the similarity. So you can see how they can take this strange jump that can cause a lot of confusion in cultivation. But here you can also see that their scans, again, are very, very similar. And so if we look at all three of these, the Rhipsalis rhombia Dave, the second one I did purchase as Rhipsalis elliptica, and this third one, I don't remember what those cuttings were labeled, but you can see how similar all their scans are, and that in this scenario doesn't just indicate to me that they're the same species, but having examined these plants and grown them in my collection for long enough, they are the same clone of the same plant. Because one of the interesting things that I found out is doing this was that every single Rhipsalis that I scanned seemed to have a unique scan, almost like human fingerprint. And then here we have a completely and totally normal Rhipsalis elliptica. And this one, it does produce those like long basal shoots, very similar to the Rhipsalis rhombia Dave. But again, this is just a completely and totally typical Rhipsalis elliptica. You never would have looked at it and thought that there was anything strange about it. Not when it was young, not as it is matured. It is just very, very typical for Rhipsalis elliptica. You can see the little pink fruit that's there. You can see the little yellow flowers. This one I received as Rhipsalis olivifera, and this one's kind of interesting because I heard not even really secondhand, but more like third hand, that there is the possibility that the botanist who described Rhipsalis olivifera identified this plant as Rhipsalis olivifera. And I don't know how any of that really went down or anything, but I placed it here because when I scanned the plant, the scan of the plant seems to indicate a very strong relationship with the Rhipsalis elliptica that I scanned. And to me, morphologically, this plant does look like Rhipsalis elliptica. Florally, it also looks like Rhipsalis elliptica. But again, I'm not a botanist, and so it's like I'm not really qualified to argue with a botanist, especially not the botanist who actually is responsible for initially describing Rhipsalis olivifera. That would be silly. But I still placed it here because this is where the scan seemed to indicate that it belonged. And who knows really, like I've read some texts that have indicated that Rhipsalis olivifera and Rhipsalis elliptica are quite similar plants, but I think that a lot of the herbarium sheets that I've seen and everything, even the holotype, there's like a holotype sheet and an isotype sheet for Rhipsalis olivifera. And I've looked at some of the herbarium sheets for things that were sampled in the phylogenetic studies, and they look quite distinct from this. So, I'm not really entirely sure in terms of the scans, like other than the fact that I have a large enough collection to scan, like this is just kind of my observations from doing these scans. It doesn't necessarily mean that it's completely and totally fact. Like this could potentially actually be Rhipsalis olivifera, and maybe the scan is just weird. Here's a Rhipsalis elliptica that is kind of interesting. This one seems to be some kind of a rather miniature form of Rhipsalis elliptica. It's quite interesting. You can even see like the flowers are a little bit interesting. The ovaries on the flowers of this are really pale. The flowers are quite small. And I think this might actually be what Alice Calvente proposed as a species, as the new species, Rhipsalis elliptica, subspecies microflora. But I've never seen the fruit on this plant to be able to confirm that, because apparently the fruit on what she proposed is supposed to be white. And the plant fell under a whole bunch of different names. So it seems like botanists typically tried to identify the plant as being Rhipsalis oblonga. In her thesis, she had taken a sample of this Rhipsalis oblonga, and it showed that it was more closely related to Rhipsalis elliptica, and that's why she had proposed that species, that subspecies of Rhipsalis elliptica. But here you can see the scan, again, looks very much like a normal Rhipsalis elliptica, only smaller, which was kind of interesting. But the plant itself is smaller, so I wouldn't think that that would be that surprising. But you can also see it has very typical looking Rhipsalis elliptica flowers, maybe just smaller. And then here we see a Rhipsalis elliptica that this plant is actually in more of a juvenile form right now, so it's quite small. I don't think that this is like subspecies microflora or anything. I just think that this is a juvenile plant. So it's like way to add even more confusion into Rhipsalis elliptica. But you can see the scan here of this one particularly looks quite similar to the scans of these. Just smaller. So again, a very typical one, a plant that you would never really look at and argue was anything other than Rhipsalis elliptica. And then we're jumping into the macranthus here. So we have Rhipsalis macranthiform raiorum, and you can see how different this scan is from the scans of Rhipsalis elliptica immediately. Like you can see that there's like a resemblance, but like not a very strong one. And so here we see Rhipsalis macranthiform kerbergii, and you can see how these do have a similar scan. And then we have this one, which is kind of confusing and annoying. This one floats around in cultivation a lot and ends up under the name Rhipsalis sulcata. A true Rhipsalis sulcata has very, very long branch segments. Like when I say long, I mean really long, like longer than your forearm. Like they're extremely long. So they can produce some that are a little bit short here and there sometimes, but the typical growth pattern of Rhipsalis sulcata is to produce very long branches. And this plant produces rather short branches that are shorter than the length of your hand. So there's a distinct difference there. And this particular Rhipsalis is also confusing because the texture, it's not very textured, it's very smooth. And the flowers, they are a little bit different from like your classic Rhipsalis macranthiform kerbergii, this one. They are a little bit different, and they do seem to have a kind of yellow pinky cast. But when the flowers are examined of this plant up against your typical Rhipsalis macrantha, florally, they have a stronger similarity to Rhipsalis macrantha than to anything else. So because this plant has typically 3 to 4 angled branch segments, it's placed in Rhipsalis macranthiform kerbergii. Whether or not that is technically where it belongs, I'm not sure. But what's interesting is that in a little bit, we'll see a plant that scans very similar to this one. So here we see Rhipsalis macranthiform macrantha. And again, we can see that there are similarities here. And then here we see another one, and we can see the similarities there. And then we see this one, where this one has a smoother epidermis, and we can see that that one scans closer to that kind of unusual form kerbergii. So very interesting. It's also just really interesting to see like, oh gosh, these plants actually kind of scan in a similar way. So I think you could really use these scans to help with identification. I don't think that you could use scans like this to identify most species. There are some cases where I don't think that's true. In some cases, I think that you could take one look at a scan like this and be like, I know exactly what that is. But most of the time, you need to have both a scan and the plant. So it can potentially help with very confusing plants to identify. So here we see Rhipsalis rhombia. And again, we're jumping into a different little tree here. So this plant is interesting because you can see how spread, how dispersed the stomata are, which is kind of like, huh? So we take the jump from this to here, we can see the stomata is pretty dispersed. And then we look at this one, I got under the name Rhipsalis occidentalis. And initially when I saw it, I thought, oh, that looks really promising. Like what I saw were like stems that were shaped like this and like this. But as this plant has started to grow out, I've become more and more concerned and been like, I think that might actually just be a Rhipsalis gobeliana that was grown under different enough conditions that it looked like it was possibly Rhipsalis occidentalis. But this scan that I took here is indicating to me that this is scanning more closely to Rhipsalis rhombia. So I think it's probably just Rhipsalis gobeliana. And here we can see what a scan of Rhipsalis gobeliana looks like. And again, like you can see, these are pretty dispersed. And I won't know until the plant actually flowers and everything. So I won't exactly be able to confirm 100% until I actually see it flower. And then we see this Rhipsalis affinis ewaldiana. And this plant has been really difficult because I think that it is different enough and weird enough that it can start growing like in a really, really strange way that is kind of, it seems atypical for Rhipsalis ewaldiana to grow. However, the scans actually indicate to me that not only are these, is it very closely related to Rhipsalis ewaldiana, but I would, at this point, after seeing the scans, seeing the flowers, seeing the growth habit, I would literally just call it Rhipsalis ewaldiana. So it's not that I think that it is that closely related to Rhipsalis ewaldiana. I think it just is Rhipsalis ewaldiana. I mean, look at this scan and this scan. Like there's no real difference here. And then we look at this Rhipsalis ewaldiana, which this one produces pink fruit. And this Rhipsalis ewaldiana is very, very similar to what the holotype, where the original description for Rhipsalis ewaldiana came from. And this clone might actually even be a holotype clone. So you can see how all these are scanning in a very, very similar way. Like there's no real confusion. They obviously are all the same species, Rhipsalis ewaldiana. And then we see Rhipsalis cuneata. This plant did not survive in my collection. It actually, I think it died very slowly over time. And I still had a piece of the dead branch. So I scanned it just to see what it would look like. And you can see that you can scan dead materials. However, you lose a lot of the texture detail, like in between the stomata, but you can still kind of make out the stomata density, the kind of pattern. So you can see the little stomata here and you can kind of make that out. And it scans look like it was probably Rhipsalis cuneata subspecies australis, but I don't know that for sure really. But you can see that there is a similarity there, even when you're looking at the stomatal pattern between dried materials and fresh materials. And then we see Rhipsalis cuneata, subspecies cuneata. This plant came to me as subspecies australis, but I think somewhere along the way, something must have happened. Like, the cutting may have been taken from the wrong plant or something. And you can see that the pattern is quite similar. It is maybe just more dispersed, which is the kind of thing that I think that you would expect to see. Like it was very similar, the same kind of thing that we saw with Rhipsalis elliptica. When we saw like the kind of larger ones, they had the same pattern or a similar pattern, and it was just larger. And then we see Rhipsalis occidentalis. This is definitely a clone of Rhipsalis occidentalis. And you can see the similarity here, but where I really see a stronger similarity is between subspecies australis and Rhipsalis occidentalis here. And it might just be, again, something about the size because they're about the same size. But you can also see a strong similarity between just the general shape of them, which Rhipsalis occidentalis and Rhipsalis cuneata were synonymized by Ralph Bauer, but for some strange reason when the phylogenetic studies happened and these plants were all sampled, they never really decided that they were the same species. So maybe there was something that was different enough that they saw there that they were like, no, they are still distinct species. So in any case, they are pretty closely related. I think that you can see that by the scans. You can see that morphologically, and you can even see that florally. And then we have Rhipsalis oblonga, and you can see between Rhipsalis oblonga and Rhipsalis occidentalis here, you can see that there is a strong relationship. Between all of these, you can see that there is a strong relationship in their scans. They do all look quite similar. Here's another one. And then we have a last one. And I thought this one was kind of interesting because to me, this scan of this Rhipsalis oblonga, this is very obviously Rhipsalis oblonga, but this is where the scans might actually get a little bit confusing because in this specific scenario, if I was to just look at this scan alone and not actually have the plant material, I would think that this scan looked very similar to some of the scans of Rhipsalis elliptica. And so I thought that was kind of interesting. And so that kind of harkens back to the Rhipsalis olivifera that I placed with the Rhipsalis elliptica, that the scan looked very similar to Rhipsalis elliptica, but that perhaps is misplaced. And then we see Rhipsalis barthlotii. Now, this plant and this plant, these two exact clones of these plants would have been materials that Nadja Kova had used to sample in her thesis. Both of these clones would have been in the botanical garden labeled as Rhipsalis oblonga, but when she sampled them and looked at them and she said, hey, these are two different species. And so then we ended up with Rhipsalis barthlotii. But you can see how these scans are quite similar. And then we have this guy, which this guy was kind of hard to identify, mostly because it came to me under the name Rhipsalis rhombia. And sometimes when a plant comes under a wrong name, particularly a Rhipsalis, if it even bears some resemblance to that species, you have to wait for it to flower. And because in a juvenile state, this plant had some resemblance to some of the Rhipsalis rhombias that I had to wait. And as this plant started to grow out, it started to look more and more and more like it was Rhipsalis crispata. And then as it continued to grow, you can kind of actually see the stages of growth here. You can see these little tiny stems back there. And then you can see them get larger here. And then you can see that they all of a sudden turn into this shape. And then they turn into this shape. And then all of a sudden we start seeing how they are completely different looking at this stage. They look really strange. They look very undulating on the edges. They're very wavy. They're very narrowed at the base versus back here where they didn't look like that. So this is a really cool example of both a juvenile and adult form of a Rhipsalis species and phenotypic plasticity. How it might change under different growing conditions. This is a really, really cool example of that. It finally flowered. And when I saw the flower, I immediately was like, okay, like that looks to be Rhipsalis crispata. And so sometimes that can get kind of confusing because you may see this growth down here and think, oh, that's got to be Rhipsalis oblonga, right? Because it's got that undulating, wavy growth. But most all these species that are in this group, and even a bunch of other ones, are capable of producing that kind of undulating growth. That's a terrible identifying characteristic for Rhipsalis. It's not something that shouldn't be taken into account because what causes it is more of the thinness of the stem. So it's not something that shouldn't be looked at. It shouldn't be weighted as that being a solid identifying characteristic without looking at a bunch of other factors with it. But anyway, you can see how the stomata are arranged here. They look very similar to that. And then if we look at a very typical Rhipsalis crispata, and this is Rhipsalis crispata. This plant, I see this plant kind of out there under a bunch of different names, including Rhipsalis crispa marginata. But actually, guys, Rhipsalis crispa marginata probably looked a little bit more like this or a little bit more like this. Like, as a matter of fact, there are Like Alice Calvente has tried to synonymize Rhipsalis bartholdii and Rhipsalis crispa marginata. So the plants must actually be quite similar in appearance. This plant is like very robust. It has these like very, very thick, prominent midribs on the stems. And the only oddity about this plant is that it's a little bit contorted, the stems. So it actually is a little bit monstrous. Like the stems are a little bit twisted and contorted, and like you can see it in the midribs when you look at them. They're like kind of twisted up, and they're wavy and going in a bunch of different directions. But it's actually quite large and quite robust. And it is really and truly historically even what was sketched in old botanical sketches as Rhipsalis crispata. So it's not questionable at all as to what this plant was. I think we've always known that this is Rhipsalis crispata. And when we go back to this plant and we kind of start looking at some of these stems like this, we can kind of start seeing some of that resemblance there. But the thing that is obvious about these two plants is that they are very obviously different clones of the same species. And so that is interesting and confusing because that can happen in cultivation where you have different clones like this and people will give them different names because they don't believe that they could be the same species just because their stems have morphological variability. What is not so variable is their flowers, though. These two plants have very, very similar flowers. And then we have this one, which this apparently originated from Shelby Gardens. You know, once when I was doing research, I saw this exact plant for sale. It was in some kind of like local newsletter or something like that for sale under the name Rhipsalis crispata. But I've never, ever seen this plant in any kind of real botanical text. I have, however, seen herbarium sheets of Rhipsalis crispata that look very similar to this. And this is another case where things can be kind of confusing. So florally, I've seen the flowers of this and they have a very strong resemblance to Rhipsalis crispata. The stems of this plant, they have a strong resemblance to the stems of Rhipsalis crispata, the previous one that we looked at. The only difference is they don't have that deep lobing. However, that deep lobing kind of thing, that's something that is like super impacted by growing conditions. It's like, I've seen all kinds of crazy clones of all these different plants. And like when you see Rhipsalis elliptica, sometimes you see that it doesn't have any kind of lobing on the edges of it. And sometimes it has much deeper lobing. And so it's the same kind of thing here. And so I think that this is very obviously a different clone. I do think it's Rhipsalis crispata. I have not seen the fruit of this plant to be able to confirm that, but I genuinely do believe that this is Rhipsalis crispata. Also, in some old botanical texts, they had described Rhipsalis crispata as looking very similar to Epiphyllum, the species Epiphyllum. And what's really interesting is that I saw this plant once presented to a botanist, and the botanist immediately was like, oh, that looks like it's probably some kind of an epiphyllum. And so I thought that was kind of interesting because a lot of things about this plant fit that it is Rhipsalis crispata. And at a bare minimum, we could just say that it is extremely closely related to Rhipsalis crispata. So you can see in the scans too, these three plants have quite similar scans. So I'm willing to bet all three of them are just Rhipsalis crispata. And then we're going to take a jump into Rhipsalis olivifera. So this is another one where the scan was taken from deceased materials. Unfortunately, this plant died very tragically in my collection. However, this is a very well-known clone of Rhipsalis olivifera at this point. This plant is not really under question as to whether or not it's Rhipsalis olivifera. It's Rhipsalis olivifera. And you can kind of see a little bit of an interesting thing here because you can see this stem has, you know, like a lot more areoles on the side of it versus this stem. It has quite a bit less. And these stems were actually quite a bit smaller than this one. So right off the bat, you could see that there was some variability that was happening here. And then we see this one, and the scan of this one looks kind of similar to that one. And again, with this is kind of interesting because you can see the relationship here of how all these plants could have evolved from the same common ancestor ages and ages back. Like you can see that there is some similarity here. And you can also see in this scan a similarity with particularly Rhipsalis elliptica. And so again, we have that one that I placed over there in Rhipsalis elliptica because the scan looked so much more similar, but it might not be correctly placed. Even though all these plants, like this plant and this plant, you can see how just from this cutting, they look like different plants. And then we come up on this one, which again, even here, this looks like a different plant, but this is also apparently Rhipsalis olivifera. One thing that I will say about this is that this stem on this plant does have a resemblance to the stem on this plant. And these are just cuttings, so it's kind of hard to really make some of that out. But even so, I think you can probably still see that these do look pretty different in their scans. And it's tough to say whether or not the stomatal, like I would say, it's hard sometimes because of the background noise, and there's a lot of background noise on this plant. Also, I don't know how long these plants were grown under the same growing conditions. What I do know is that none of the growth happened in my collection. So these aren't scans from plants that I've had in my collection. And so there might be some variability due to that as well. But I mean, really interesting at the same time. And then we see this one, which this one right here and this one are very, very similar. They scan very, very similarly. So I'd definitely be willing to say that these two plants appear to be pretty closely related. And this particular plant, like I've seen some of the other growth. It's not indicative of the cutting here, but I've seen some of the other growth on this plant and everything. And when compared up against the holotype herbarium sheet and one of the specimen that Alice Calvente used in her dissertation, this plant actually looks quite similar. I'm not showing the plant because they're not pictures of my plant, but one of the other things that I want to point out that I think is very interesting here is that while these two plants seem to be scanning with a very close relationship, you can see how deep the lobing is on the edge of these stems and how it's non-existent here. Again, that's something that is really, really variable within the same species. So that's not something that I look at and I'm like, no, it can't be. I think what's more interesting is the lower amount of areoles on these branches. I think that's kind of interesting when compared to other kind of similar species. I think it was interesting because it seems like there's less. And here we see this very confusing plant. So this is an example of like a Rhipsalis that I found that it was an unidentified species that apparently came from a botanical garden. It's highly unlikely that it's some kind of like new species. And I know that seems like some kind of a surprise because it's like, well, I've never seen a Rhipsalis that looks anything like that. But just because we've never seen a Rhipsalis that looks anything like that doesn't mean that it's a brand new species all the time. We kind of have to see the flowers and the fruit and everything first, because obviously I think like through this we can see that their stems are highly variable. So the only times that I've ever seen any Rhipsalis that looked anything like this was there was a botanical sketch of Rhipsalis elliptica that bared some resemblance to this. And there was some pictures of Rhipsalis cuneata subspecies australis that I saw that looked like this, kind of similar to this. They weren't exactly, but I placed this here even though some of the scans, like this scan particularly, looks like some of the scans of the kind of odd Rhipsalis elliptica, the one that we were calling Finis at the beginning. These scans actually bear a stronger resemblance to those than really to anything else. But I also put it here just because maybe it's wishful thinking. I don't know. I put it here because I could also kind of see that there was some resemblance here too. And so again, might kind of go back to the thing like maybe Rhipsalis elliptica and Rhipsalis olivifera, like they might have kind of similar scans sometimes. I'm not entirely sure. And then we have this little weirdo. This is particularly difficult. The scan of this does not quite look like anything else really that I scanned. And this plant came to me as Rhipsalis olivifera, and it came to me from like a collector who is very unlikely to mislabel things. And so it kind of surprised me when I saw this plant because I thought there is no way that plant could be Rhipsalis olivifera. It's so small. Like the stems on this plant are really tiny. So then I kind of looked at it and I was like, well, maybe it's like some kind of weird Rhipsalis oblonga, or like perhaps some kind of like Rhipsalis cuneata, you know. So I think I was like trying to figure out exactly what this plant was, thinking that it couldn't be Rhipsalis olivifera. And, you know, the scan of this just made it extremely hard to place. So I decided to place it where it was labeled. And I mean, maybe I was wrong. And maybe Rhipsalis olivifera really can be like this very strange little plant, because in some ways I do see some similarity in the stems between this one particularly and this one. And so... But I've also seen this plant, and I know that this stem on this plant is actually atypical for how the plant grows. But sometimes there's kind of an oddity where, like, you can see how sometimes some plants will have like two different stem shapes, and then you'll have another plant that's the same species that predominantly has one of those stem shapes, and another plant of the same species that predominantly has the other stem shape. And so that can be kind of weird because it seems like they can produce particular stem shapes that fit their environment, and that can get kind of weird sometimes. So, you know, I think that the same thing was true here. Like this stem had kind of a resemblance to some of the stems that were on this plant. And at the same time, I went back and looked at the original pictures of where I collected this plant from. And when I looked at the original pictures, honestly, their original pictures of their plant look nothing like what my plant looks like now. And so again, an example of phenotypic plasticity where this plant's appearance is way different now in my collection than it is in their collection. And even in their collection, it's a very odd looking plant. So I don't quite know what this is. Like in some way, this scan does bear some resemblance to some of the scans of Rhipsalis oblonga. So this one will be particularly interesting if it ever flowers to figure out what it is, especially because these stems are so oddly small. I've seen a botanical sketch of Rhipsalis oblonga that does kind of look like this. So highly confused about this plant. But this is where I placed it because it seemed to maybe kind of resemble this scan a little bit, but honestly, didn't really resemble any scans. And then we come into a group that's really interesting. So we had that big group where they all evolved from the same common ancestor. And these scans are particularly interesting to me. So here we have a scan of Rhipsalis russellii. This was not grown in my collection. The scan here was taken from this cutting. And then we have one that the scan was taken grown in my collection. And I think that what we can see about these two is they're obviously a little bit different. And so they're obviously different clones of the same species, but they're similar enough that you can probably make out that they're the same species. And then we have Rhipsalis serioides. And so this was kind of surprising to me about some of these. You can see the similarity, the relation between these plants, even though their stems are so totally different looking. I mean, these are really, really different looking species, but even their scans seem to say these are really closely related. And then we have Rhipsalis agaduensis. Now, Rhipsalis agaduensis is very closely related to Pachyptera. They were in a sister clade together, and some botanists had even synonymized them. And so when we look at the scans between these, we can see that there is a pretty strong similarity in their scans. As a matter of fact, all 4 of these species have very strong similarities in their scans, and their scans appear to be very unique. They seem to have much larger sort of stomata, and they are like closer together. Overall, the stomatal density is lower for these species just because the stomata is so much bigger. It's really, really, really interesting here. And then we see another one where I had put this one as Rhipsalis species affinis Pachyptera. This had produced one flower one time, and the flower was very confusing because it looked like Rhipsalis Pachyptera, but it was kind of smaller. And so I think I had labeled this as affinis Pachyptera, but I actually think it's just Pachyptera. And here we get to see something that's really cool because I have enough Rhipsalis Pachyptera in my collection that we really, really get to see and understand clonal variation while still being the same species. And here we see another Rhipsalis Pachyptera. And I mean, you can see the variability in the stems here. You can see that they're very closely related. They are different clones, but their scans they're strikingly similar for Rhipsalis Pachyptera. So I think that these scans can really, really help to differentiate between things like Rhipsalis Pachyptera and Rhipsalis elliptica. And here we have a Rhipsalis Pachyptera that was actually kind of a smaller plant. The stems on the plant are a little bit smaller. Very obvious Rhipsalis Pachyptera flowers. The flowers are just a little bit smaller. And so here we can see the scans are really similar, but like how we have seen in some of the other species, the pattern is just a little bit smaller. But you can still make out that it's very obviously the same pattern. Here we see another one. Once again, we see the same kind of pattern. This kind of blew my mind when I saw this. And then here we see this one, which is really interesting. This plant I've had for a while. It has never flowered. And it came with the name Rhipsalis confer with elliptica, which means compare against elliptica. They didn't think that it was the same species as elliptica, but thought that it should be compared against it. And the label here is wrong on this. It's actually ISI 9616 and not 9610. And I bought this from Rhipsalis tropiflora. So let's go to the Rhipsalis tropiflora website and see if there is any kind of characters that are there that are going to help us identify this a little bit better. Okay, so here if we go here, we can immediately see that they have some picture of some buds. And if we look at these flower buds, we can immediately see that the ovary is short, like very, very short. Just like when we had initially looked at the pictures of a comparison between Rhipsalis elliptica and Rhipsalis Pachyptera. And we can see this kind of like round bulbous fruit up here. We can see how these are like, they're just very short ovaries. And I think that this would actually indicate to me that this plant is most likely Rhipsalis Pachyptera, just a clone of Rhipsalis Pachyptera. And I think that the scan was very, very helpful in saying that was probably true without actually having to see the flowers or the fruit. I mean, you would still wait for the flowers and the fruit, of course, but I still think that that was a very good indicator that this plant was probably Rhipsalis Pachyptera and not Rhipsalis elliptica, especially in a case like this, where the plant actually does have a fairly strong resemblance to Rhipsalis elliptica under certain conditions. You can see it's also confusing because it has that reddish coloration to the stems. that is something that I think has always been really confusing, because I think that it's commonly misunderstood that Rhipsalis pachyptera has green stems and Rhipsalis elliptica has red stems. These are terrible identifying characteristics, just really not good characteristics. Rhipsalis elliptica does appear to be more likely to have reddish stems in cultivation, but that kind of thing is just really down to betulins and like just how the species was grown in the wild, where it was collected, all of those kinds of things. So it's a highly unreliable characteristic. And then we come into another branch where we come into Rhipsalis grandiflora and Rhipsalis pidieri, that little tree. And so here we can see these scans are very different. Obviously, these scans are wildly different from any of the scans that we've looked at thus far. And these are interesting because the plant is so textured that you can't really pick up the stomata from these scans very well. All you can really do is pick up the channeling in the epidermis, and you can only kind of sort of make out some stomata sometimes. And so here's another scan of a different clone, and you can only kind of sort of see the stomata in there. Sometimes it's not very clear. But here is a third clone where we can finally really clearly make out what Rhipsalis grandiflora is all about. And we see these channels, and then we see the stomata that is sitting inside of these channels. Really cool scan. And if we look at Rhipsalis pidieri, we see a very similar scan. And so I think I was always really surprised when they were like Rhipsalis pidieri and Rhipsalis grandiflora are pretty closely related. I think I was always like, are they though? Like, I can see the similarities sometimes, but I think it's also kind of confusing. And, obviously without those phylogenetic studies to tell us this always would have been confusing because we were basing it off of like floral characters in terms of the flower ovary was actually kind of immersed in Rhipsalis pidieri, and it produced trichomes that is more indicative of it having been Rhipsalis flacosa. So that's kind of interesting, where I think Rhipsalis pidieri was playing some kind of a trick on us. But actually it was just independent evolution. Like they would have evolved that way due to some kind of similar circumstances. We're going to have to start zooming through these. Otherwise this video is going to be 3 hours long. So we're coming into the subgenus Rhipsalis now, and you can see the species that we're going to be seeing here. And I'm only really going to stop and talk about these when there's something that is like really, really fascinating. Otherwise, we're just sort of looking at the scans and seeing the similarities. So here we see Rhipsalis sulcata. And the phylogenetic studies told us that Rhipsalis sulcata and Rhipsalis teres are very closely related. As a matter of fact, they really could not actually tell them apart genetically. So here we can see the scan of Rhipsalis sulcata. And again, you see these very long branches that form on Rhipsalis sulcata. Again, they're literally longer than like your forearm. And here we see Rhipsalis teres form heteroclada. And I think that there is a difference in the depth of the texture of these two plants that were scanned here, but you can still see the similarity there. That was really shocking to me. Here we see another Rhipsalis teres form heteroclada. And then we see another Rhipsalis teres form heteroclada. And you can see how these scans look similar to Rhipsalis sulcata, which is very surprising. Now, I do want to say that this plant particularly, I want to say that when I was working on another project, I had to do a ton of research, and Rhipsalis teres form heteroclada and Rhipsalis teres form teres have historically always been very, very confusing. Their botanical descriptions are confusing. It's very hard to tell. Like sometimes it seems like the descriptions were backwards. And because of that, what I did was I went to all of those phylogenetic studies and I actually started pulling all of the plants that were sampled. Now, Nadja Koratkova's dissertation is the one that is actually helpful with this, mostly because her dissertation, she sampled very clearly plants that were labeled as forms from the botanical garden. And I was able to actually find several of those clones of plants that she sampled to figure out exactly what botanists were sampling as Rhipsalis terrariform heteroclada and what they were sampling as Rhipsalis terrariform terras to get a much better idea of the plants that we probably should be calling that. And what I ended up finding out from doing that is that there's actually a lot of variability in Rhipsalis terrariform heteroclada, a lot more than you would have thought. And actually, all of those kind of chunky boys, whether they're upright like this or whether they're pendant like this, all of the kind of chunky boys that have the terrace flowers are technically form heteroclada. So that was kind of interesting. There's a ton of variability there. And surprisingly so, in that some of the samples of plants that I saw in Rhipsalis terrariform heteroclada that were sampled, it was interesting because they did share a striking resemblance with some of the plants that you see in Rhipsalis bassifera subspecies Mauritiana particularly. So I thought that was interesting. And then we see Rhipsalis terrariform prismatica, and you can see a similarity in the scans here. And we see another Rhipsalis terrariform prismatica. And then we see a Rhipsalis terrariform terras. And so I felt like doing that kind of in-depth research was extremely helpful to be able to differentiate form terras and form heteroclada. And so we can see that these actually have quite narrow stem segments in comparison. And so there's a lot of them. This particular plant I had a really hard time trying to identify because it's never flowered. And I think I always thought that it was terras, but I just didn't really have any way to really confirm that. But the scans of this plant indicate to me that it is actually terrariform terras and not a bassifera. And so last we see Rhipsalis terrariform capilliformis, and we can see that there is a relation here. We can see the similarity. And again, I mean, actually the scan is really, really similar to form terras. And then we see these, which is another thing that is really, really interesting. Now, there's two plants that I scanned here. This plant in particular is the one that is very, very easy to see. There were plants that were apparently collected by Horich in Costa Rica, and he collected them as Rhipsalis similari initially. And it's very strange that you would find Rhipsalis terras growing in Costa Rica to begin with. Rhipsalis similari was actually a name that was already taken, and it was applied specifically to Rhipsalis serioscula. That was reduced under Rhipsalis serioscula ages and ages ago, probably by Wilhelm Barthelot in 1995. What ended up happening, though, is that these plants ended up falling under Rhipsalis terras. And in my collection, when I saw these plants, I was like, there is something oddly different. And so I thought it was maybe just a very unique clone of Rhipsalis terrariform capilliformis. Because you can see here another one of these plants, they have very, very thin branch segments. They have very tiny flowers. The flowers have a striking resemblance to the flowers of form capilliformis, but they never quite exactly fit, but that's okay just because there's like tons of variability there. However, when I was doing all of that research, I ended up finding out that Nadja Korotkova had actually sampled one of these plants. And when she sampled it, it did show that there was a real possibility that this was perhaps an actual subspecies of terras. But she said there had to be more research to confirm that. So here we just call this Rhipsalis species Aphanis terras. Like, it is obviously very closely related and most closely related to Rhipsalis terrariform capilliformis. So very, very interesting, very unique, curious plant. And it does grow in this funny way where it creates these like long basal stems and then creates these like weird pom-poms at the end. It's a frequent kind of habit that it has. And so it's kind of easy to see how they may have thought that it was Rhipsalis serioscula initially. And when Horich wrote an article and kind of described this plant, he described it as bearing kind of a resemblance to the growth pattern of Hadioris salicornioides form cylindrica. And it does. When you put the plants, those two species up next to each other, you're like, wow, that really does. Like you can see it here. It really does have a striking resemblance to that. So very, very, very interesting. Also, just as kind of a funny side note, at one time, I do believe that they had considered introducing the name Rhipsalis teres form costaricensis, or maybe it was Rhipsalis teres subspecies costaricensis for this. Anyway, the name costaricensis seems to have been something that was considered, but I don't believe that that was ever any kind of a published thing, but kind of funny. And we see Rhipsalis bassifera subspecies horrida, and you can see that there was kind of a similarity in some of the scans here between horrida and teres. And then you see this is another one that I scanned, and this one was just a crested one. So I just wanted to see out of curiosity, was there anything different? Here we see Rhipsalis bassifera subspecies erythrocarpa. And so again, we can see a similarity here. And then we see Rhipsalis bassifera subspecies mauritiana. This is a clone that was collected from Gabon, Africa. And so this one is actually a really interesting, beautiful clone of this plant. The flowers, the tips of the petals have kind of like a purplish or a reddish coloration to them, as do the flower ovaries. So it's just a really striking clone. And then we see Rhipsalis bassifera subspecies mauritiana. This is another one. This one's very confusing. It's out in cultivation. It has a kind of an odd resemblance to Rhipsalis teres, but you can see the scans here telling us that this is a bassifera. And just out of curiosity, the same plant, this is actually a juvenile version of this exact same plant. And you can see the juvenile stems, they have a lot of like soft little spines on them that eventually go away and the plant takes on a completely different appearance. And so again, it's Rhipsalis playing tricks on us. But here we can see the scan, even though it was kind of distorted, you can still make out that the scan is not really that different in the juvenile versus the adult growth. And then we see another one where this one is also subspecies mauritiana, and you can see this one has a very textured surface. And then we see another one. This one is the cultivar Ferrari. And a last one here. This one's actually one of my favorites just because I like the flower ovaries. You can kind of make it out here. The flower ovaries have like a reddish underside to them. I just think it's kind of interesting looking. And then we see Rhipsalis bassifera subspecies bassifera. There's a lot more variability in Rhipsalis bassifera than one would think. A lot of it. And it's actually really kind of interesting, some of the variability here. This one probably would have gone under the name Rhipsalis quellenbembensis at some point in its history. And what's very curious about this particular clone of Rhipsalis bassifera subspecies bassifera is that it has a tendency to produce these odd sort of like orangey, unripe fruit. So, but it is also subject to growing conditions. So this plant, I got cuttings of this plant from someone whose plant always produces these kind of unripe, orangey, pinkish fruit, and then it ripens to white. And so she sent me cuttings, and in my collection, when I had it in my cabinet upstairs where it wasn't receiving any kind of natural sunlight, but artificial light, it did not actually seem to be strong enough to produce that kind of flush on the unripe fruit. You can see that there is like one fruit that's on the plant that managed to do that. And on occasion, you can actually see the kind of blush on the green fruit. So I thought it was kind of interesting that the only reason why this plant was producing that kind of coloration on the fruit was sun exposure. And then we see Rhipsalis bassifera subspecies bassifera, one that is very typical that we all know and recognize. And so you can see the similarity in all of those scans. And then we see another one, and I could not actually tell the difference between this one whether this was Rhipsalis bassifera subspecies bassifera or whether this was Rhipsalis schaeferi. And you can see the scan of Rhipsalis schaeferi, and so that would indicate to me that this is probably Rhipsalis bassifera subspecies bassifera. And so interesting, and another one in my collection that scanned like it was Rhipsalis schaeferi and has the flower buds and everything like it's Rhipsalis schaeferi. So very interesting. And then we come to Rhipsalis mesembryanthemoides. And we can see here this scan was interesting to me because it has a really strong resemblance to some of the scans of Rhipsalis ewaldiana. And that may just be because of the texture that's on the stems. But you can kind of make out a little bit underneath that texture, the channels. Like you can kind of see the channels that are actually on there. So maybe not like ridiculously different from Rhipsalis schaeferi, but the floral characters between Rhipsalis mesembryanthemoides and Rhipsalis schaeferi are actually very, very similar. So kind of interesting, kind of weird, like a weird placement for this Rhipsalis. And let's be honest, this Rhipsalis is just weird among all the Rhipsalis. So, and now we're going to jump into another part of the tree. And we're going to be looking at here at Goniorhipsalis, the subspecies Goniorhipsalis. And one thing that happened here is that we found out that Rhipsalis pentaptera, Rhipsalis pachecoleonis subspecies cadenulata, and Rhipsalis lindberghiana seem to form a clade together, having all evolved from the same common ancestor. And that Rhipsalis flagelliformis and Rhipsalis pachecoleonis subspecies pachecoleonis form a clade and evolved from the same common ancestor. And that would probably indicate that subspecies cadenulata and subspecies pachecoleonis are not as closely related as they appeared to have been initially. And so here we can see the scan of Rhipsalis pentaptera, and you can see where the stomata is supposed to be here. So you can see there, but it doesn't seem like this was like it couldn't quite pick up the eye of the stomata really in most cases, but you can still get an idea of what the scan looked like. And then we see subspecies cadenulata, and you can see a similarity in these scans. You can also see how these scans are actually different from any of the other scans that we've looked at so far. And I think that this is one thing that was really interesting, that when Alice Calvente initially, like that paper was written, when she sampled the plants, she took plants that were specifically from all of these different subgenus and looked at them in that light. So it kind of interesting that all of these subgenus seem to have kind of indicators for what belongs with them. So here we see a Rhipsalis lindberghiana. This one didn't have a very textured surface. And then we see another one which does have a very textured surface, and we see these channels, the same kind of channels that we would have seen in Rhipsalis grandiflora and Rhipsalis pidieri. And so the channels are here on this one, I'm sure, but I'm sure that it's just smooth enough that we can't actually pick it up, but you would be able to see it if you did a proper epidermal peel. So interesting. And then we see Rhipsalis flagelliformis. We see one clone of it, and then we see a second clone of it. And interestingly, notice that there's no channels here. We don't see any channels on this one, really. So I thought that was kind of interesting. And you see a very clear difference in the stomatal pattern here. So kind of cool. And then we see Rhipsalis pachecoleonis, subspecies pachecoleonis. And this particular clone it has the ability to produce totally white flowers and the ability to produce the kind of purpley or fuchsia-y tinted flowers that are indicative of this species, but that's down to growing conditions. And it has a tendency to produce white fruit instead of pink fruit. And so that's also kind of an interesting thing, but you can see how these scans have a strong resemblance to Rhipsalis flagelliformis. But they all do kind of have a resemblance to each other as well. And then here's another one, and this is a different clone. This one produces pink fruit and pink flowers, always, no matter what conditions it's grown under. And so you can see You can kind of see that this one actually does kind of have channels. You can see the channels and then the stomata in the channels. So kind of interesting. It seems like it was kind of dependent on what exactly was scanned. I think that in one of these scans, I scanned this kind of spiny growth. Like you might think that's juvenile growth, but this species has a tendency to produce that growth all the way through into adulthood. So it's not something that it only does as a juvenile. And then here we see another one, which is kind of interesting. This is a cultivar. I'm not so sure about the name, if it's Burl Max like that or Burl Marx, or I'm not entirely sure. And initially, this plant was labeled as Rhipsalis flagelliformis because it apparently had very strong characteristics that resembled Rhipsalis flagelliformis when it was young. But more recently, it's been examined closer. It's flowered, it's produced fruit, and it very much seems to be Rhipsalis pacheco-leonis, subspecies pacheco-leonis. And so here we can see what the scan looks like of this. And I thought this was really interesting because the scan of this plant looks more like Rhipsalis lindberghiana. You can see it's got those channels and the stomata in the channels and stuff. Something that is here, but is actually kind of faint. So kind of interesting. And then we're jumping into another subgenus here. So we're jumping into Epilogogonium. And all we have here is Rhipsalis paradoxa. And so here we see subspecies paradoxa and subspecies septentrionalis. And you can see they look similar, but not exactly identical. And then we're going to jump into Erythro-Rhipsalis. And you can see how these are formed, like Pilocarpa and Campus potoana, each other's closest relatives, even though that seems super bizarre. And then there's confusion. I don't know where Pulchra, Aurea, Burchellii are actually supposed to be here. So I think I just kind of fudged this into whatever. And then we see Siriuscula, Jungari, Ormondoi, and Clavata. And some of those are fudged again because there's not actually a complete phylogenetic tree. Here we see Rhipsalis pilocarpa, and when we look at Rhipsalis campos potoana, indeed, we see very similar scans between these two. And then we see Rhipsalis pulchra. I took multiple scans of these because, A, they were kind of hard to scan, and in a lot of ways, the scans actually resemble the scans of Rhipsalis flacosa. What's interesting is that Nadja Korikova kind of told us in her dissertation that there was perhaps like some kind of an ancient hybridization event between a species that was in this subgenus and a species that was in the subgenus where Rhipsalis flacosa was placed. And so we see like bizarre characteristics of some of the plants that are over there and Rhipsalis pulchra. And so in their scans too, they seem a little bit funny to be placed here. But here is the two scans of the same plant. And then here's a second clone, and there are some scans of this one. And we can kind of make out the little channels on one of those scans. And then here we see a last one. And this one was particularly interesting. Here we can see these two scans. Interesting because it seems to really stand out like a lot. And actually this plant is a plant that was kind of confusing because it was in my basement. It never actually had a species declaration. It was something that was just tucked away and not something that I really paid that much attention to. And when I was doing these scans and I scanned it, I was like, whoa, that's super weird. And this is something that I don't know what it is. Like I initially thought that it was just like some kind of Rhipsalis bassifera or Rhipsalis teres or something. The cutting was too small to identify it. Eventually, I kind of thought that it was a Rhipsalis bassifera because of the way that it was growing. But obviously the scans here are telling me, no, that's something totally different. So I actually asked the source of where this plant came from, like, hey, have you ever seen this plant flower or fruit or anything? And as it turns out, they had, and they sent me pictures of it. And it turns out it's the most beautiful clone of Rhipsalis pulchra I have ever seen. So I had this amazing plant just tucked away in my basement, which I think is kind of ridiculous, really. But I also thought it was kind of interesting because of the variability in these scans between these three plants. And I'm not exactly sure what to make of that, but two of these scan very similarly. And actually, this clone AH690 looks very similar to AH781 florally and everything. So really interesting to see that variation in the scans. And then we see a scan of Rhipsalis aurea, and you can see it very much looks like it's placed correctly in this subgenus. And we see Rhipsalis burchellii. Again, we can see the similarity in these scans. Rhipsalis serioscula, once again, very strong similarity in the scans. And we see Rhipsalis jungeri, and now Rhipsalis jungeri is looking a little different. Here we see a Rhipsalis species that I picked up from Andes orchids in the United States. And the moment that I saw this plant, I knew that there was something different about it. There was something different about the way that it was growing, just everything. Like, it was a very small plant, but I knew I had never seen this species before in person, except for as a young plant. So I knew it had to either be Rhipsalis jungeri or Rhipsalis ormondoi. And I bought another one and it came with fruit. And when I looked at the fruit, I was like, oh, that looks like Rhipsalis jungeri. But then there's some problems here. Here's Rhipsalis ormondoi. And let's go back again, Rhipsalis jungeri. So Alice Calvente, when she did her thesis, she had sampled Rhipsalis ormondoi and Rhipsalis jungeri. It turns out that she could not actually get Rhipsalis ormondoi to resolve in its own distinct branch as its own like species. It just wouldn't resolve in the right way. And it turns out that she proposed the idea that Rhipsalis ormondoi was actually a hybrid of Rhipsalis jungeri and Rhipsalis clavata, or possibly, and that's part of what was wrong there. And I have since seen some very strange oddities with plants of Rhipsalis ormondoi. Like, the original description for Rhipsalis ormondoi was very, very strange because it kept telling us that it had white fruit. And then a botanist got it to fruit and everything in cultivation and amended the original description to tell us that it had yellow fruit, that the fruit was like a golden yellow color. And there are pictures of it and everything, and it does indeed look like golden yellow. And since then, I have actually seen the same botanist had other collections of this and shared pictures of them. And it was kind of crazy because the fruit looked kind of pink with an undertone of green. And that was really interesting because Rhipsalis jungeri has fruit that has, it's green sometimes. Sometimes when it's ripe, it's green. And sometimes when it's ripe, it's kind of like a weird reddish color. And the whole thing is very confusing. And so I think that what we can see is that Rhipsalis ormondoi probably has a lot of variability in its fruit and everything because of the possibility of it actually being a hybrid. Is it a recent hybrid? Probably not. So it's probably something that's like an older hybrid in, out in the wild or whatever, and has been collected as this distinct species. So really wildly interesting. And so that brings me back to the Andes orchids plant, where I still cannot actually identify it for that reason. To me, the plant, like you can see here, actually looks more like Rhipsalis jungeri. And now that the plant is older, it looks even more like Rhipsalis jungeri. But I still have to wait for the thing to flower and fruit and everything now because of this rather strange fact. And here we see Rhipsalis clavata, and we can see again, these scans look very much like they belong in this subgenus. And we see this Rhipsalis clavata. This is one of my favorites just because the plant flushes red, and because grown in higher light, it has like these really cool short stubby stems, which is interesting because it explains why this at one time would have been classified under Hadiora. It's very, very fascinating and cool looking plant. And then we see Rhipsalis clavata form delicatula. And what's interesting is between delicatula and clavata, we can see that the only real difference in the scans appears to be the size of the pattern, and it's very minute. And here we're jumping into another subgenus now. This is the last subgenus, and we see this is Rhipsalis. And we see Rhipsalis trigonoides. Now, Rhipsalis trigonoides, of course, is the plant that we used to know as Rhipsalis trigona. And so we can see here that it has obviously something very different from most of what we have seen. The closest thing that we've seen that potentially looks like this would have been Rhipsalis pulchra. And again, there seems to be some kind of tie between Rhipsalis pulchra and this subgenus. And now we see Rhipsalis dismillis dismillis, and you can see that there is a very clear, distinct difference between the scans of these two plants, indicating that they are probably not exactly the same species. So I think that Najakorakova proposed that they would probably have to look really, really closely at the plants here and determine whether or not they were in fact subspecies. The two plants share very, very strong similarities, both morphologically and florally, and even in their fruit. And then we see there's going to be some confusing things that are here. So here we see a plant that Lombardi described as Rhipsalis spinescens. And since I've seen some publication where it was proposed that this is actually a juvenile form of Rhipsalis dismillis epiphyllanthoides. Now, I've never seen the flower of this plant to really know. And some of this is really confusing because this would have been the holotype for what was described as Rhipsalis spinescens. This would have been the paratype. And so a paratype is when a second collection is made and the description is amended to include the characters of the second plant that was found. And obviously these two seem pretty different. And so it's kind of confusing because I don't know that this plant really matures into this. I kind of don't think so, but the scans do tell us one thing about these. There is a relation here. They are related. They both belong clearly in this subgenus. And they clearly seem to have some kind of close relation to each other. And they don't quite seem to be as closely related to Rhipsalis dismillis. At least that's what the scan is saying. But like we saw with Rhipsalis pulchra, perhaps something a little bit different is going on there. Perhaps they are more closely related. It's confusing. Especially because technically Rhipsalis spinescens, this one, the paratype, would have been reduced under Rhipsalis dismillis dismillis. And this plant has yellow flowers. It's got these yellow flowers. The back sides are reddish. It doesn't really have so many petals. So it's kind of interesting. And assuming that this plant has the same flower, that seems like a very distinct and different flower from Rhipsalis dismillis dismillis. So something kind of funny is going on here. And hopefully someday they do a little more research about this subgenus particularly, and some of the reductions that were made here. And then we see these plants, which are in cultivation, and these are very confusing because they seem to have a very strange difference in their morphology. It seems like they start out one way as juveniles, and as they mature, they start to look more and more and more similar to some of the Rhipsalis flacosa or to some of the Rhipsalis dismillis dismillis. And it's very interesting because these plants were technically reduced under Rhipsalis dismillis dismillis in 1995 by Wilhelm Barthelot. And originally, these plants may or may not have gone under the name Rhipsalis rigida. Reading through all of the descriptions, there were several plants that were actually reduced under Rhipsalis dismillis dismillis, and they all shared the same kind of interesting characteristics. They all had straw yellow flowers, and all of them had kind of the backside of the flower was kind of a reddish color. They have a little bit of a different growth pattern here, where sometimes they can look really crazy like this. So what we see here is a ton of juvenile growth. And then only at the base of the plant do we actually see some mature growth. And this particular plant I picked up from Tropiflora under the name Rhipsalis grandiflora. However, I have a friend who has a plant that looks almost identical to this. And I asked him about it, and he said that he was given the plant from one of the curators at Tropiflora, and that it was originally from Rainbow Gardens and originally labeled as Rhipsalis grandiflora. These scans are very similar, telling me that these two are probably the same or very, very closely related based off of everything that I've seen in my collection and all of the looking at, staring at all of these plants and all their little tiny details and everything. It indicates to me that these are the same species. Here we see, I took another scan of this just out of curiosity. I kept taking scans of these just to kind of see, how similar are they really? And this was a cutting from the plant that my friend had that he sent me, and I scanned to see what I could see here. Now, this is very interesting because obviously his growing conditions are different from mine. And what I can see here is that there is some room for differences in these scans. So either these plants are the exact same plant, which evidence suggests that they should be the exact same clone of the exact same plant, or something is amiss. But I think that they are the same clone of the exact same plant, and we're just seeing some minor differences, which we've seen in scans of some of the other plants too. When I've taken multiple scans, sometimes there's minor differences in the scan. But what was very interesting about the difference between my scans and the scan of this plant is that these scans share a strong resemblance with Rhipsalis tucumanensis, which is kind of interesting. So, we can see here that there was less stomata and it was kind of more spread out. And we see less stomata and it's more spread out here, but we also kind of see maybe a little bit of different texture. Maybe there's a little bit of channeling there. Kind of interesting. And I took 2 scans just to be sure. And here we can see that there is more stomatal density there, but we can still see that something is a little funky between his plant and my plant. And that we might have a really hard time placing them if minor differences in growing conditions can cause something like that. And here we see a Rhipsalis flacosa, and I don't know what subspecies this is. And here we see another one, and this one is supposed to be subspecies Pulvin ingera. And you can see again where all these scans are pretty similar. So here we see one that is apparently from Bonn, and it's 5751. And this would be a known clone of Rhipsalis flacosa subspecies Pulvin ingera. This plant is almost dead in my collection, sadly. No matter what, I just could not get this plant to root and grow. It was really a terrible experience. Then we see another one, which Rhipsalis flacosa AH711, and it says possibly subspecies Pulvin ingera. The reason why it says possibly is because apparently Rhipsalis flacosa subspecies Pulvin ingera and Rhipsalis flacosa subspecies flacosa can be so similar, and because their growing area overlaps, that you can't really tell them apart very well. And so apparently they've even been found growing in the same place, which I'm going to guess is probably going to be something to look at much closer as to whether or not they are really different subspecies. Their growing areas, they integrate. It's something that they've always known about these, but that's why this says possibly, because it was a plant that was collected in one of the overlaps and because it was very difficult to really tell apart. And here we see another one, and it's the same kind of scenario. And we see a last one. And this one was a cutting from a friend that this one carried the name Rhipsalis gibberula at one point, apparently. And that was something that was reduced under Rhipsalis flacosa subspecies Pulvin ingera. And you can see all the similarities in the scans here of all these. And then the last one that we get to, which does seem to have a different scan, at least the one scan that I took. So here we see Rhipsalis flacosa subspecies hohanaensis. And you can see the stomata just looks like it's more spread out here. And then we see Rhipsalis dismilis forum epiphyllanthoides. And you can see this one's got like the short growth. And it's interesting with this because you can see how the stomata is more spread out here. And you can even see how that does actually share kind of a resemblance here with this. And so that's pretty interesting. But apparently it is not as closely related as originally thought. And so here we see this, and then I took a second scan of this juvenile growth to see just how much did this actually resemble the little Rhipsalis spinense that was over here, this little guy. And they seem to be related, obviously, like they're obviously all placed correctly in the correct subgenus together, but not very clear as to whether or not they were completely reduced correctly. To me, when I look at these and I look at the plants and I look at the flowers and everything, I feel like they were not reduced correctly. They're not correlated to the right species, or there's more species in this subgenus that we don't know about just yet. And so maybe someday all these plants will be sampled and we'll kind of find out. Not these specific plants. I mean plants that were reduced in this subgenus. And then we see Rhipsalis chucuminensis. And so you can see like how this chucuminensis has a very similar scan to my friend's that was probably like this Rhipsalis rigida at one point. You can see how there was a strong similarity here. So this was kind of head scratching to me where I was like, interesting. But it'll be interesting, too, to rescan his plant when it starts producing new growth in my collection and see if it actually starts looking like my plant does. So here we see another Rhipsalis chucuminensis, a different clone. And you can see this one has more texture on it. And you can see the stomata, again, is like very, very spread out, very, very dispersed and larger. And this is another clone, a third clone in my collection. And this one has flowers that are kind of more pinkish. And you can see, again, the same kind of texture. And you can see the stomata behind that texture. Here is another scan that I took of the exact same clone. You can see the flowers. They're kind of like those pinky flowers. And again, you can kind of see very strong texture, and the stomata is very spread out. And then a last one that I have, which I believe is probably Rhipsalis chucuminensis. And then we see Rhipsalis nevis armandii, form megalantha. This scan is a little bit distorted. The reason why this scan is distorted is because the plant is kind of dehydrated, and taking this with dehydrated materials like that can actually cause some weird things with the scans. It can make the texture appear very deep or cause distortions like this. So it's just a little bit distorted. Here is another one that's in my collection, Rhipsalis nevis armandii, form megalantha. And you can see how it's got like these channels, these big channels, big stomata. And then we see one that is an intermediate in my collection, which very strongly appears to just be right smack in the middle between like normal clones that I have, smaller size clones of Rhipsalis nevis armandii, form nevis armandii, and larger Rhipsalis nevis armandii, form megalanthas. This is not unusual. And actually, I've read some criticism of the fact that these were even declared as forms due to the size difference. That's probably not something that should have been done. And it's kind of interesting because going over many of the other plants, we can see that there is a size difference between some of the other plants, and they don't have form declarations. That would be something that I would think would not be necessarily be a great characteristic for a form. And then here you see a completely normal clone, and you can see it's got these channels again. Same channels, same kind of stomata thing. Another clone. Another clone, another clone. Yeah, I actually have even more clones of this plant that I have not scanned. That's because I have like a real interesting thing where I really, really like Rhipsalis nevis armandii. But you can see this is actually a scan of one that has yellow stamen, and this is a scan of one that has pink stamen, and you can see that they're pretty similar here. And then there's a couple of plants that I assume that this is Rhipsalis nevis armandii, form nevis armandii, but I have never seen it flower. And the reason why I have to say I assume is I don't even have a label on this plant, so I don't even know where it came from. And so here we see another one. This is one that I picked up that was very bizarre because the flower has a strong resemblance to Rhipsalis nevis armandii, but the stamen is not just pink, it's red, like deep, deep red, more like Rhipsalis puniceodiscus. The growth on this plant is very weird, like sometimes it's determinate, sometimes it's indeterminate. Added to that, I have seen the fruit of this plant, and the fruit of this plant is pink. It is not orange, so the plant cannot actually be Rhipsalis puniceodiscus. And that means that this plant is either just kind of an unusual clone of Rhipsalis nevis armandii, form nevis armandii, or it is in fact one of these fabled hybrids. And here we see another one. Now, this is a plant that I picked up from Cacti and Hage. I imported, and this one was labeled as Rhipsalis puniceodiscus. And this is another one that does not quite have the right growth pattern. It does not quite have the right flowers. Like, it's weird. I've yet to see the fruit on this plant. And so this is a plant that I don't quite know what it is. The flowers are a little bit strange, but the flowers isn't really what's bothering me about it. So you can see a lot of times when it comes to clonal variation, like we saw when we looked at Rhipsalis pachyptera, how we could have like those great big flowers with lots of petals and then the smaller ones that had less petals. That's not unusual. So it wouldn't be that weird for Rhipsalis puniceodiscus to produce flowers that have way more petals and then something like this, which has way less petals. That's not what's strange here. What's strange here is the overall habit of the plant and how it does not actually look like Rhipsalis puniceodiscus. So this is another one where I have to kind of like wait around and, try to get it to fruit and everything before I can really make any kind of determination about what exactly this is, or if this is potentially another one of these fabled hybrids between Rhipsalis puniceodiscus and Rhipsalis nevis armandii. Mind you, like Rhipsalis hybrids, they had never been reported. Hybrids did not exist. Like, this, the first time that anything was ever reported that was a potential hybrid was when Nigel Taylor wrote a paper and basically brought up a possible hybrid between Rhipsalis puniceodiscus and Rhipsalis nevis armandii. Now, since then, obviously, like Nadja Korikova has brought up in her dissertation, the idea of ancient hybrids, but ancient hybrids are not the same as hybrids. Like, this would have been a hybridization event that caused speciation. So that's something that is different. And then when we come to Alice Calvente and her research indicating that Rhipsalis ormondoi was potentially a hybrid. And so I think that we can also look at something like Rhipsalis aurea and contemplate whether or not that is a potential hybrid between something that was in Rhipsalis and something that was in Hadiora. Obviously because of the yellow coloring of the flowers, that is kind of a curious thing. We also know that there is the possibility of Hadiora selacornioides and Rhipsalis serioscula having overlapped in their growing environment in nature and producing a hybrid. And that's really interesting because I think I can never remember how to say the like hybrid moniker or whatever, but that's an intergeneric hybrid. And that does exist. Like, there is a paper where in Japan, they actually proved that they could hybridize these. So there really is a hybrid between Hadiora selacornioides and Rhipsalis serioscula. So there's possibilities that are kind of coming up that are happening there of potential hybrids. That doesn't mean that every time you see a Rhipsalis that is a little bit different or unusual that there was a hybrid there. So much clonal variation that is happening that to assume that there's a hybrid would probably be a really big mistake. Here we see Rhipsalis puniceodiscus. This one has yellow stamen. And then we see another Rhipsalis puniceodiscus, and this one has the kind of like reddish stamen. And there's nothing unusual or weird about either of these plants in any way, shape, or form. They're not growing strangely or anything. You can see that it has long indeterminate growth. You can see that the branching pattern is subacrotonic, totally normal looking. And this one has the same kind of growth pattern, same branching pattern, everything's totally normal looking. And you can see the funness in the scans with the big long channels and the stomata here. So really cool scans. And then this was probably my favorite scan, this scan of Rhipsalis hauleri, just because it was so clear and attractive looking with the long channels and the stomata and everything. And so just really, really interesting because these two, Rhipsalis puniceodiscus and Rhipsalis hauleri, are pretty closely related. And so you can see that similarity in their scans. And the last one is this little Rhipsalis hauleri that is potentially white. This was like some weird thing that was found where it was like a really strange anomaly with a little tiny white flower that really only kind of resembled Rhipsalis hauleri, and the fruit and everything looks like Rhipsalis hauleri fruit. It's just that the flower is kind of bizarre and whitish. And the flower shape is a little bit odd for Rhipsalis hauleri. So just a kind of interesting thing, but I thought that I would scan this and just see what I would see, what did it look like, and this is what it looked like. And it was interesting because, like, obviously these are pretty closely related, but it was also kind of interesting to me because this scan looked really, really, really similar to the scan of this plant, which I thought was interesting. Like, kind of weird, right? Anyway, I mean, I can't really draw any kind of conclusion about that because I know those plants to be two totally different plants in appearance. I just think that it was kind of interesting that the scans were so similar. Anyway, I hope that you've enjoyed this and, maybe got some curiosity or insights there. I know that this is pretty long and it was a pretty wild thing, and there's a lot of scans and there's a lot of practice and pattern recognition between some of them and everything. But, I hope it was insightful in some way. Thank you for watching. And as always, happy cacti growing.